here is our final genetics video. It is going to be on dihybrid crosses. I am going to tell you this now. It's going to be the longest video, as dihybrid crosses do take a fair amount of time to go ahead and complete. So the first thing that we need to go ahead and talk about is something called linked genes. And linked genes are genes that are located close to each other on a chromosome. So an example of a linked gene here would be our A and our B, or A and our B. So we could go ahead and say that they are linked because they're very close to each other. So when alleles are linked, they move together during meiosis to create gametes. All right. So there's going to be very few gametes made that do not have A and B traits, or A and B traits. Odds of having A and B, or A and lowercase b, are low. All right, so even in the crossing over process, they are going to go ahead and stay together. All right. If you're curious about linked genes, definitely go ahead and take genetics your junior or senior year with Miss Ricciardi. For our purposes here, we're going to be worried about unlinked genes. So we have two pairs of homologous chromosomes, and these genes are located far from each other on the chromosome, or on separate chromosomes entirely. These genes move independently of each other during meiosis. So when meiosis occurs, crossing over can go ahead and occur. There will be just as many gametes that have A on the chromosome as allele, say D. all right? Because they're all moving separately from each other. So this was something that Mendel did not actually know about when he was going ahead and doing crosses. So we're going to examine this a little bit further. So Mendel's other law. So Mendel took true breeding strains with two different traits each. All right. And so he went ahead and he looked at white flowers with round seeds, right? Which the genotype for that would be white flowers would be lowercase f, and round seeds would be capital R, all right? And crossed with a purple flower with wrinkled seeds. So he ended up having, purple was capital F, and wrinkled seeds was lowercase r. So the resultant plants that he ended up getting were these purple flowered round seeded plants. So I denoted round seeds by coloring them in green. So the phenotype for his offspring, for his F1, was purple flowers with round seeds. All right, so he ended up having two of these. So our purple flowers were capital F, lowercase f, capital R, lowercase r. And we got that by if you did two monohybrid Punnett squares, all right, and you isolated, only did the one for flower color and then only did the one for the type of seed. And when he ended up crossing them, all right, so this was the parent generation. He had white flowers with round seeds, and then he crossed and he had purple flowers with wrinkled seeds. He got all purple flowers with round seeds. And then he let them self-pollinate. And he ended up seeing that he got some with purple flowers and round seeds, some with purple flowers and wrinkled seeds. Notice I didn't color them in. Some with white flowers and round seeds, and some with white flowers and wrinkled seeds. So when he looked at that, out of 16 different plants, he got 9 out of 16 were purple and round, 3 out of 16 were purple and wrinkled, 3 out of 16 were white and round, and 1 out of 16 were white and flowered. So how did he figure this out? On the next page, we're going to do a giant Punnett square. Let's go ahead and review our law of independent assortment really quickly. Mendel discovered that each allele was inherited independently, all right? Inheriting flower color didn't impact seed shape. All right. The thing that he didn't know at this time is these traits are unlinked and on separate chromosomes. That's something that we know now. If I was you, I would go ahead and I would color in your purple and your round seeds just to go ahead and help you. And we're going to come back to this page in a little bit. So based upon what Mendel had been working with, all right, we need to talk about which traits are dominant and which traits are recessive. So which traits or alleles are dominant? We know we have capital F for purple, and we have capital R for round, and we have lowercase f for white, we have lowercase r for wrinkled. So the first thing that Mendel went ahead and he did was he figured out the phenotypes were all purple round in his F1 generation. And that's what he crossed to end up with his ratio of 9 to 3 to 3 to 1. And so the genotypes were all capital R, F, lowercase f, capital R, lowercase r. This is capital F, lowercase f, capital R, lowercase r. So now come the gametes. And so if you remember foiling from math class, all right, you're going to put the capital with the capital, all right, and then this capital with the lowercase. So we're going to have a capital F, capital R, a capital F, a lowercase r, and we're going to have a lowercase f with a capital R and a lowercase f with a lowercase r. And that would be the exact same thing for this one. So outside, inside, outside, outside, inside, 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 outside. Okay? So now let's go ahead and let's take those gametes and put them across the top and put them across the side. So we know that we have purple, round. We know that we have purple, round. All right, so we have capital F, capital R, capital F, lowercase r. I'm following the exact same convention here. Lowercase f, capital R, lowercase f, lowercase r. And then the gametes would have been exactly the same for this one. Capital F, capital R, capital F, lowercase r, capital F, lowercase f, lowercase r. Okay, so now we need to go ahead and start filling in our Punnett square, which is very similar to how you previously filled in Punnett squares. You're always going to make sure that you have your capital letter prior to your lowercase letters. There's no lowercase letters here. Capital F, capital F, capital R, lowercase r, capital F, lowercase f, capital R, capital R, capital F, lowercase f, capital R, lowercase r. And so on and so forth for all of your Punnett square boxes. This is why I told you this was going to take a little bit longer to go ahead and do. So the genotype is rarely discussed when we talk about dihybrid problems. We really discuss the phenotype of both of the genes. The phenotypes, we have purple round. We have purple wrinkled. We have white round. And we have white wrinkled. And we know that we have 9 of these, 3, 3, and 1. So I am going to go ahead and I'm going to take 4 different colored pencils at the current time. And for the purple round, I'm going to say that it's this light blue aqua color. Okay, so we're going to find all of our purple round plants. So that means we need at least 1 capital F and 1 capital R. And I'm going to shade those in. So these are all our purple and round pea plants. Skip that one. Purple, skip this one. Purple and round again. Purple and round again. Skip, skip. Purple and round. And the rest of these are going to be white. All right. So now we're going to take our next color, my purple and wrinkled. So I would need to have a capital F, but 2 lowercase r's. So capital F, 2 lowercase r's. Okay. And I have a capital F, lowercase f, but 2 lowercase r's. So we're purple and wrinkled again. And then a capital F, lowercase f, but 2 lowercase r's gives us that a wrinkled purple plant. So the next one is going to be a white flowering plant with round seeds. So I'm going to denote that in yellow. Okay, so we know for our white plant, we need to have 2 lowercase f's, and our round seeds, we need to have at least 1 capital R. Here we have our 2 lowercase f's and capital R, capital R. All right. And then we have our last, which is a plant that shows both recessive traits. They are white and they are wrinkled. And that is right here. So if we go back to our previous page, we could go ahead and see where our ratios came from. I can go ahead and color in this box. It's the same as this one right here. And then our white flowers with our round seeds was yellow. Our purple flowers with our wrinkled seeds we made pink. And then our purple flowers with our round seeds, so our dominant is aqua. All right, so that's how you do a dihybrid cross when you're going ahead and you are looking at 2 genes that are unlinked, so they are inherited separately from each other during meiosis. And this is your last genetics video.